I'm Joseph Hamilton, a PhD student in Sarah Tabrizi's group at UCL, and today I'll be presenting our mechanism of how FAN1 controls CAG repeat expansion in HD by two functions, MLH1 retention and nuclease activity. So we know that higher somatic expansion correlates with an earlier age of onset and faster progression in HD, linking it to the pathogenic process. We also know that expansion relies on the mismatch repair pathway. This is involved in the detection and resolution of DNA loopouts. In this instance, they're occurring in the CAG repeat tract of Huntington exon 1. This involves the MUT-S beta complex composed of MSH3 and MSH2, as well as a MUT-L endonuclease complex, the core of which is always MLH1. We know that FAN1 is inhibiting the process of somatic expansion, but we weren't sure how. Obviously, this entire pathway is an interesting and attractive therapeutic target as we would modify age of onset as well as progression. Several GWAS studies over the past few years have linked variants in mismatch repair factors to age of onset in HD, including MLH1 and MSH3, but also identified this gene FAN1. So FAN1 is an endonuclease involved in the resolution of interstrand crosslinks, and it's also shown relevance to somatic expansion and trinucleotide disorders. Knockdown of FAN1 in HD cell models increases somatic expansion, reflecting FAN1 knockout mice in Fragile X. So at 6 months, you can see much more expansion, particularly in the striatum in FAN1 knockout. Mechanistically, it's been shown that FAN1 is able to bind MLH1, but it's not been followed up in a HD context. And our hypothesis was that FAN1 is binding MLH1, pulling it out of the mismatch repair pathway and reducing somatic expansion. The main cell line we use is this FAN1 knockout U20 cell line. It has a flip-in locus, so we're able to reconstitute the cells with a GFP tagged FAN1 variant, and we also co-transduce with a Huntington exon 1 construct. This allows us to look at somatic expansion of the CAG repeat in the context of the FAN1 variant, and also allows us to look at the DNA repair interactome and perform functional assays. Initially, we wanted to confirm that FAN1 indeed bound MLH1 in our systems, so we have two HD cell systems rather, and if you pull down FAN1, you indeed pull down MLH1, and the reverse is true. You pull down MLH1, you get FAN1. We confirmed this by GFP trap pull down of FAN1 in the U20 cells and also in two independent HD mouse models. We have an ongoing collaboration looking at cross-linking immunoprecipitation mass spectrometry in HD lymphoblastoids in FAN1 MYC expressing HEC cells. And through this, we're able to identify a cluster of 3 FAN1 MLH1 cross-links within the unstructured domains of the respective proteins. This fed forward into our structure function studies as we believed then that the MLH1 binding capacity is at the N-terminus of FAN1. We made many constructs. We have the 1 to 3, 4, 9 here, which is just that N-terminus, no other functional domains. And we have the full length 73 to 3, 4, 9 deletion construct, just missing these residues here, but having intact this nuclease domain at the C-terminus. GFP trap pull down of FAN1 shows that 1 to 3, 4, 9 is sufficient to bind MLH1 with the same affinity as wild-type full-length FAN1, whereas deletion of 73, 3, 4, 9 residues is sufficient to abrogate MLH1 binding, suggesting this is where that function lies. We also performed an MMC assay, which is a measure of the nuclease activity of FAN1, and as you can see, the 73 to 3, 4, 9 mutant acts just like wild-type. In looking at somatic expansion, the 1 to 3, 4, 9 acted like a wild type, whereas deletion of the 73, 3, 4, 9 residues increased somatic expansion between that of wild type and a full FAN1 knockout, suggesting that MLH1 binding and somatic expansion are mechanistically linked. Note that as an intermediate level, however, so something else is also controlling residual expansion activity. We then performed a conservation analysis on the N-terminus of FAN1 and identified this SPIF motif here. This resembles an MLH1 interacting protein or MIP box motif found in known MLH1 interactors such as MSH3. To this end, we looked at its role in MLH1 binding by performing SDM with alanine substitutions within the motif and also a control Q123A just outside. Indeed, mutation of the residues in the motif abrogated or almost completely abolished MLH1 binding, whereas the Q123A had no effect. And linking it back to somatic expansion, mutation of the SPIF motif only accelerated expansion between wild type and a full FAN1 knockout, indicating they're linked mechanistically. So we wanted to look at whether FAN1 is directly competing with MSH3 for MLH1. We performed MLH1 IPs, first in U20 cells, expressing full-length FAN1 and a FAN1 knockout, And you can see the expression of FAN1 decreases this MSH3-MLH1 interaction. We corroborated it in 125Q medium spiny neurons that are FAN1 knockdown. And if you knock down FAN1, you increase that proportion of MSH3 bound to MLH1, indicating that complexes have that competitive nature. We then performed a 6TG assay in the U20 cell. And essentially, if you mutate the SPIF motif with the Y128A or F129A, you decrease viability in the 6TG assay compared to wild type, which is indicative of increased mass match repair occurring. So altogether, this leads to our conclusion that FAN1 reduces CAG repeat expansion by pulling MLH1 out of mismatch repair and reducing that as activity. But it doesn't answer the question of why do SPIF mutations or the 73 to 349 deletion mutant not expand like FAN1 knockout cells? So we looked at FAN1's canonical function as a nuclease. On top of the MLH1 binding mutation, we performed D960A mutation, which nullifies the FAN1's nuclease domain. And this is us characterizing it here. And most importantly, removing the MLH1 binding capacity and the nuclease capacity of FAN1 produced a somatic expansion rate equivalent to a FAN1 knockout, suggesting these two mechanisms account for FAN1's ability to stabilize the repeat. This is an illustration of our mechanism here, that the two mechanisms are involved in MLH1 sequestration and FAN1's nuclease. And the promotion of the FAN1 MLH1 binding interaction therefore may modify age of onset or progression. We're following this up in more physiological endogenous HD systems like our IPS systems as well. So thanks very much for listening. I'd like to thank our funders as well as everyone involved in the project, Takes a Village, specifically Rob Gould, who spearheaded this work.